as a title. I thought it read a bit like a Daily Mail headline. So this is a sort of more scientific and possibly more realistic and possibly less appealing title that I won't even bother reading out, but that's what I'll be talking about. So the work we're doing is, this is a Michael Swan building, level 3, that's the structural biology part. And in the past year or so, as this project has developed, we've developed nice links with Keith Matthews and Achim, who have been exceptionally helpful in guiding us towards the animal testing side of things. And when I look at this photo, everybody knows Arthur Seat. I think this is predominantly Edinburgh audience. But I always think the last time this was active was about 340 million years ago. And it was really about 300 million years ago that the group of trypanosomatids, kinetoplastids that we're interested in, or we have been doing structural work on, diverged. And so that becomes a bit relevant later on. I don't know if anybody ever looks at Arthur Seat and thinks that, but that's my... Actually, it's a sort of excuse because my zoology, biology, because we've been doing structural biology on kinetoplastids, I tend to mix up leishmania, trypanosomes, and we've got a whole lot of structural information. And the structural results I'll be showing are sometimes from leishmania, and sometimes from T. cruzi, and sometimes from T. brucei. But for the pathway that we're interested in, they're all very, very similar. So forgive my occasional lapses of being exact in terms of biology. But we are very interested in this particular disease, human African trypanomyces, where we've got some money to do some actual drug discovery work. And as nobody else is talking on the hat, then just a couple of brief background bits of information. So the number of people infected actually is dropping considerably. Sub-Saharan Africa, obviously, the main species are rhodesiensis and gambiensis, are the infect humans. Large numbers of people at risk, but only about 8,000 reported hat cases each year, and that seems to be dropping. But it goes up and down with political unrest. So the trypanosome parasites, they're spread by the bite of a tsetse fly. And according to the font of all wisdom, which is Wikipedia, David Livingstone was the first person to link tsetse fly bites with sleeping sickness. So the tsetse fly transmits the parasites, which live in the bloodstream form in blood, free living in blood, which is a perfect environment where they get fantastic nutrition and are quite happy as long as the host survives. So once you're infected, then you've got stage 1 hat, where you get some symptoms, but after a few weeks or months, then the parasite crosses the blood-brain barrier, and then it's a really unpleasant stage of the disease, and stage 2 hat, where you get malaise and sleeping disorders and so on. Actually, the major economic impact of trypanosomiasis is with cattle. And the zoonosis, it's Congolense and Vivax are the main species that are infecting the cattle, but there is zoonosis. So eradication is going to be problematic. And the disease costs about over $1 billion a year to the African agricultural economy. So it really is a major problem. The unmet need, spectacularly, the diseases these are the available clinical drugs that are used to treat hat. And Suramin, discovered over 100 years ago, is still on the list as the drug for use for initial infection. All the other drugs are really pretty old. The current drug that's mainly used is this dual therapy, eflornithine and nefertimox, but it's still required to be given intravenously. So 4 current drugs and 2 that are up and coming. One is from a company, Synexus, in phase 1 trials, and another one, fexinidazole. There's no known mechanism, and even these new drugs, you can see that the dosage is enormous, 25 mg per kg, 200 mg per kg. And this one's even positive in the Ames test. So there is a current need for new orally effective drugs that will get into the brain. And the pathway that has been championed over the past 30 years by Paul Michels and also Linda Gilmore is the glycolytic pathway as the potential target for new drugs. And we're very lucky to have Paul Michels has joined us here in Edinburgh to work with us on this target. So the glycolytic pathway is pretty ancient. It's over 2 billion years old. Amazingly, the 10 enzymatic steps are pretty much conserved in all organisms. So the 10 enzymatic steps that require the breakdown of glucose, which is a nutrient, down to pyruvate. So in most organisms, pyruvate is then fed into the mitochondria, and you get this massive production of ATP. So you can get 10 molecules of ATP per pyruvate. This pathway here, at the final step, it's dephosphorylation of phosphoenolpyruvate to pyruvate by pyruvate kinase. That actually produces ATP. So the trypanosomes that are living in this luxurious environment of blood with millimolar amounts of glucose always available, when it's in that state, the mitochondrion is pretty much shriveled up, and it doesn't bother producing ATP with that pathway, with this organelle. It relies 100% on this 10 enzymatic steps to produce ATP. So if you can block the ATP, the parasite dies immediately. So that's the rationale of using this pathway as an interesting target to kill trypanosomes. I put this picture also of a cancer cell because, interestingly, the parallel is that cancer cells are very much dependent on glucose. They're really addicted to glucose to grow. And the reason is that all the metabolites on this pathway are used in the production of phospholipids, amino acids, nucleic acids to produce the material required for cellular growth. So the 2 enzymes that we in Edinburgh have been working on for quite a few years now are the 2 sort of major controllers in the pathway. This is phosphofructokinase, and that is the step that adds another phosphate onto fructose 6-phosphate to make fructose 1,6 bisphosphate. And then pyruvate kinase, this final step, which is producing pyruvate and ATP. And this slide actually summarizes about 10 years of quite hardcore crystallography to determine the structures and the mechanisms. So it's a pretty flashy slide. And we can see, so everything's moving, and I'll try and take you through some of the sort of details of these molecules. But there are similarities. The real gatekeepers are allosteric enzymes, so that means that they need a different molecule to activate them. They're tetrameric, so there's 4 chains, each of about 500 amino acids, in both of these, pyruvate kinase and PFK. So an important paper, important piece of work was from Paul Michels' lab, where he looked at RNAi inhibition of knockdown of different enzymes in the glycolytic pathway. And this is showing that for both PFK and PYK, that if you knock down the enzyme, then the trypanosomes are dead. And you don't need full knockdown even, you just need 50% knockdown. So that's the rationale that we're using and working on. So I'll take you through some of the architecture of these enzymes and how we're trying to use that structural information to identify new inhibitor molecules. So this is a 500 amino acid long chain, single chain, and this is a tetramer, and this is morphing an inactive and an active state. So the intermediate is just pure fiction, but the two endpoints are from X-ray crystal structures. And the active state is binding the substrate, phosphoenolpyruvate, and it sits at the bottom of this tin barrel domain here. This is the B domain that flaps up and down and traps the substrate along with ADP. And in that active site, you get transfer of a phosphate onto the ADP to make ATP and produce pyruvate. Now, the miracle of this is that 40 angstroms away, there's an effector molecule, and in the trypanosomatids, that effector molecule is fructose 2,6 bisphosphate. In mammals, it's fructose 1,6 bisphosphate. And the question is how, by binding this molecule so far away, does it activate this machine and allow this phosphotransfer to take place? And the answer is in this movie here, and we can see that what's happening is that each of the domains is, if we ignore the flapping B domains, but the main rocking motion, there's a concerted rocking motion that's going from the active to the inactive. Actually, I couldn't resist showing this. This is even flashier. So a student, Ankita Nathani, she's been working on molecular dynamics of this system to see if we can actually understand what the pathway, how the information flows from the effector site to the active site. And she showed this at a group meeting yesterday. And I thought, well, this is the active state. So we've got the 4 molecules here in its absolute active state. All the ligand binding sites are occupied. This is the fructose 2,6 bisphosphate wriggling about, but holding things together. This is the interface here between these 2 subunits and these 2 subunits, and we'll see in the next slide that interface is locked when the effector molecule binds. And then we've got these 4 active sites, each of them occupied by, in this case, because it's an X-ray crystal structure we're using, the product, so it's ATP, and oxalate is that little green thing as a mimic for the product pyruvate. So it's really trying to escape here, but it can't because it's locked in, hydrogen bonded to the magnesium ion. So it's quite important also. We tend to think of these X-ray structures as absolutely rigid. This is 80 nanoseconds in the lifetime of an active pyruvate kinase. So we're sharing it quite a number of times over. But more detailed in this transition from the inactive to the active state, when the fructose 2,6 bisphosphate binds, then it induces a flip, and we see what's happening here. These are charged residues, these aspartates and arginines. They come together and form salt bridges. So across this interface, when the fructose 2,6 bisphosphate binds, it somehow induces or traps the tetramer in an active conformation. So all the domains are moving in together and then locking across this interface by these, in fact, it's because of symmetry, 8 salt bridges. So it's really trapped. When these things rock, then at the active site, 40 angstroms away, this chain comes in. So this arginine here belongs to an adjacent chain. It comes in and firms up a pretty floppy helix here. So when the helix is floppy and in the wrong conformation, it cannot bind the substrate. But when it's in the active conformation, locked by this adjacent chain, then we see the substrate comes in. In fact, it's a product because it's in that crystal structure. But the geometry is perfect, and it's locked in place. So we've got a very nice understanding of this allosteric activation by fructose 2,6-bisphosphate in terms of gross molecular movements. So does that help us design new inhibitory molecules? So this is a picture of the active site. This is electron density with all the different ions. This is a product, ATP, and the oxalate. And we use that picture in this form. We've developed some software where we can look at molecules and say, well, what we're really trying to do, we don't want ATP there, we want an alternative molecule that is going to sit in the active site and block it. So, and it's got to be obviously unique for the parasite pyruvate kinases. So this software, Edulus, which is available to everybody, we use that to look for molecules that would have the same sort of geometry and shape as the ATP in the active site. So we want a hydrogen bond donor, 8.5, 8.8 angstroms away from a phosphate type molecule, type atom, and so on. So we've got these geometrical constraints. We've got a database of 3 million compounds, available compounds. We go in and say, what molecules have got the geometries that would match these atoms in the active site? And that's what we did. And these were the sorts of molecules that we managed to pull out. And they happen to be quite colorful. So we bought these molecules. The beauty of this database is that they're all available compounds, so you can buy them and test them. So we bought the molecules to test if they were going to inhibit the enzyme. They did inhibit the enzyme, and we co-crystallized them, so you get these beautiful colored red and blue crystals, which indeed are reminiscent of trypan blue. So essentially, what we've done is rediscover the compounds that our friend Paul Ehrlich discovered over 100 years ago. So I read Paul Ehrlich's papers, which is, I mean, it's quite spectacular to read what he did. And this was a summary of the work that he did in 1907. And what we can see is, so this is trypan red. These azides are chromophores, so they give brightly colored compounds. But his experimental technique, I don't think, would pass the BBSRC today. He worked purely on mice, infected mice, and spectacularly cured them of trypanomyces. One of the main side effects was, in these cured mice, was they were bright red, and they stayed bright red for weeks. So this is clearly not ideal for a drug company, or in most cases anyway. But what was fantastic, apart from being successful, he really was so ahead of the game. He coined the term chemotherapy, and then in this paper he suggested combination therapy. The next step was going to be combination therapy. But the mind boggles at the number of mice you would have to actually test in doing these experiments. So we obviously then thought, well, we've rediscovered this class of molecule. Let's see actually how suramin does work. The very first anti-trypanosomatid compound. And we managed to co-crystallize. So the development from trypan red was to try and get rid of the color. And so the chemists at Bayer did chemistry that removed these chromophoric groups. So we see suramin has just got amides. Now, this, if you show that to a drug company as a potential drug molecule, they would keel over laughing. It doesn't hit any of the requirements of low molecular weight, solubility, whatever, but happens to be a pretty fantastic, pretty amazing sort of molecule. So we soaked suramin into the crystal of pyruvate kinase, and indeed we managed to get the complex. And here we see how it's working. This is half of the suramin, and it's lying right on top of the site of the adenine ring of the ADP-ATP. So it's acting as an ATP-ADP inhibitor, competitive inhibitor. Now, this is highly unlikely that this is the main mechanism for suramin. It does bind to tens of enzymes, but it's got this broad these broad activities where presumably it's acting as an ATP inhibitor in a whole spectrum of enzymes that magically kills the parasites and not us. So the next step was to look for other molecules, and we luckily got a grant with the NIH people to do high-throughput screening, where we were looking for inhibitors of pyruvate kinase. And they've got this fantastic setup. They use a luciferase assay where the light is a readout. This is Anton and the NIH robot. The setup is spectacular. This robot swings around like a crazy thing. You're testing 500,000 compounds in well plates, 1,500 well plates with just tiny amounts. So everything's down. The cost of doing a run like this is just enormous. We got some hits out of that. The main hit was this saccharine analog, which looked pretty promising, but had this so-called flat structure-activity relationship. And what the mechanism for inhibition of this class of molecule was, you see, this is acting about the same, inhibiting about the same level as this molecule, and that's not inhibiting at all. And the reason for that is that it's a chemical reaction, so it's a suicide inhibitor, and it's reacting, again, close to the ATP binding site. The saccharin group here is reacting with a lysine that sits just over the ATP site. So this is, and indeed, at the same site, it's suramin. So we do have a theme that inhibiting pyruvate kinase through that route is interesting and maybe worth following up. However, we've had more luck with this part of the pathway, phosphofructokinase. Again, the allosteric mechanism should spring to life. We can see we've morphed, I'll just use this one. So this is the active site here, and this is the allosteric site. For the parasites, AMP is the only known activator. So when AMP is bound, the whole tetramer comes together. We can see these two giant helices from two different chains slide past each other and lock. They get locked by the allosteric activator. So we've got very good understanding of the mechanism and geometry of the enzyme. So our first foray into trying to find inhibitors here was with Nick Turner and Matt Nowicki, who made a series, a library based around the substrate, which is fructose 6-phosphate, adding different groups to each end here. And we got reasonable success. So with this molecule, with a dichlorophenol, we got good inhibition, low remaining activity against the enzyme, which didn't inhibit the mammalian enzymes or the auxiliary enzymes. So we got a couple of molecules that were showing reasonable activity. But then we went back to Anton and the robot for another grant, which again used a variant of the assay, which gave a light readout. And here, over 300,000 compounds screened, same setup with the plates, and we got over 1,000 hits that inhibited the enzyme to less than 50 micromolar. And when these hits were analyzed, there were some interesting families that popped up. So remember, this is our attempt with our tiny library, where we got an IC50 of 23 micromolar, ED50, 30 micromolar against the trypanosomes. The original hits here were even sub-micromolar, which is a really encouraging start. And all the families from this analysis had this interesting dichlorophenol group. So the chemists at NIH did a bit extra chemistry synthesizing around this lead molecule, and by playing around this part here was shown to be absolutely required. The phenyl group here was required, and if you tweaked this hetero ring group, then you could go from 450 nanomolar down to about 40 nanomolar. But unfortunately, there wasn't a corresponding improvement in killing the parasites. Then we managed to get one of these compounds co-crystallized to see exactly what the mechanism was for this family of molecules. And here we see, just to line it up, this is an overlay, it's not the same structure. This is where ATP is, and this is where the inhibitor lies. So it's lying close to the active site. So the substrate, fructose 6-phosphate, would be sitting around here. So this would be competitive with the substrate, but not with ATP, and that's what the kinetics show us. And that's also really important because you don't want to have to compete out ATP to get your drug in. And what we found, so when we compare structures with the mammalian enzyme, there's only 25% identity between the mammalian enzyme and the parasite PFK. And this is an overlay of the mammalian, which is in gold, and the parasite, which is in purple. And we can see there are significant differences. And in the parasite one, so this is the X-ray structure of the parasite one, we've got this fantastic pocket, which is perfectly shaped to take the dichlorophenol group. And that explains why that molecule keeps coming up. And if we overlay that on the X-ray structure of the mammalian PFK, we see that pocket is not present. So that's why we get this fantastic discrimination between binding and inhibition of the parasite compared to mammal. And that's obviously what we want for a drug. So just very briefly, this is a work that's now funded by the Wellcome Trust Seeding Drug Discovery Award, and this is where we are with the project. So we've got a team of chemists in a company called Celsia, who are very interactive, very good. They're producing about 10 compounds a week. Compounds get sent up, and we test all of them against inhibition of the PFK, the T. brucei PFK, and also against the parasites. The good ones, and these are our target values, so the better ones will get tested against cell toxicity, and then we've just begun to look at mouse studies, and obviously the goal is to show efficacy against infected mice. The stage we're at just now is essentially this structure-based drug discovery, where structures of the ligands complex guide the chemists, so that we're hopefully going down the path of producing more potent compounds. And the next step, and being helped here by Keith and collaboration with Jeremy Mottram and Simon Croft, who've developed this glowing parasite mutant, which will be very helpful indeed, because this is a study of infected mice. So we see the parasites glowing, very heavily infected mice, and then in this case treated with melarsoprol, and the infection disappears. But horribly, you get a relapse where the parasites start growing back in the brain. So this particular assay, which has just been developed, I think will help us a lot in the next 6 to 9 months work, where we'll be actually testing the compounds in mice. So I should thank particularly Paul Michels and Linda Gilmore for actually driving this work and keeping faith in this pathway for so many years, and Jackie Dornan, Ian McNay, who are working, and Nick Gray, who are the team who are working with the Seeding Drug Discovery Award. Thank you.